Today, I'm sorry, I could not join the presentation, the Dr. Lee, so Jun's Lee. So, but I understand the fact your very nice result. And also, Dr. Shu is very wonderful presentation. But I think I present about a little bit different. Our approach is the support the microbiome research. So maybe you can also maybe you can do same things to support, because this one is by the, we are culture collection. The culture collection can do this one. The other organization cannot do. Just you can imagine like that. So, and then, I'm sorry, I will skip. I'm happy to join the, back to the ACM members. So, and also I will skip the time to explain about that. The culture collection, no, not culture collection, biological resource centers is the activity, you know, everybody, 2 days we run. So, in the collection, preservation, and provision of microbial resources. And then also this one is just a support to the international or national law related to bioresources. Another one is provide technical support and databases. So today I focus to the, difficult, yes. The one is the collection, preservation, provision of microbial resources related to microbiome research. The one is that, also I want to skip first one, but a little bit I want to show you. So the one is the already previous, the previous presenter, much more very big result and effort were presented for the microbial strains isolated from microbiome, gut microbes and the soil or something like that. And also we do this one. And the second one is we apply to microbial cocktail, development of microbial cocktails, which are available for measurement reference. So, and I'm very happy because 10 days before our papers was published. This is in the paper, we explained about how to make mock community microbial cocktails for measurement reference, because this one is a method is a different, quite different from other commercialized one. So I will try to very simple explanation. Okay, and just more, it's okay, more already background, how important, and also more already Dr. Lee maybe, and then Dr. Shea already explained about how to isolate quickly, rapidly. Also, we try to do approach about the cultural mix, because we have, especially the culture collection staff knows how to identify, how to isolate so quickly. So, and also I'm very interested about the identification of Mavdi-Toffmers. So, and then we try to do like that from fecal sample and use the variety of media and so many collect colony and apply to identification using Mavdi-Toffmers. So we collected so many things. Same with this one. Yeah, like that. So, and also maybe I cannot include in my presentation. This is also another news is that 3 days before, our new identification system was commercialized. So in the 20th also, so and then I cannot put in my presentation. That is very interesting one is that maybe everybody has the Mavdi-Toffmers. Someone has the German machine is a Brukas. Another one is a Japanese Shimazu. And another one is Chinese Mavdi or something like that, European Mavdi. But our product, commercialized one, can be used for variable data from every Mavdi-Toffmers machine. So, and then, so later I will send, if you are interested, so I will give the website. So it's a not so expensive one. So it one is that the very interesting one is that if we use the system by the system in the cultural mix, we can identify 85,000 species, which are including unculturable microorganisms. So, and then if you, today I don't talk, just I want to introduce we also develop the new system to support cultural mix. But today is I talk about, yes, so also maybe today our colleague talk about our system, how to distribute the microorganisms. So this is a new system for, we improve the efficiency of distribution processes. This is not only microbiome cultures, but also we are doing all of the NBRC cultures we are doing. Like this is yes. This is the automated stock is a maybe also in Thailand also there is same things, but also in our collection the automatic packing system was also developed. So maybe in your future please visit to our institute so and then please look so that so and then we should think about the quickly identify, quickly isolate, and also quickly distribute in the no mistake or something like that. So, and we are every time thinking about that. And the skip that today's my talk is about the microbial cocktails. Okay. Just slowly I talk. Okay. Yes, どこだっけ? Yes, microbial analysis, high throughput DNA sequencing, enable details characterization on complex microbial community and is a cornerstone of today's microbiome research. Everybody knows. But the problem is that I'm sorry, this is difficult. Problem is differences in protocol between laboratories often lead to poor reproducibility and limited comparability of microbiome data across studies. That is a problem. So more than 10 years, more everybody knows that this is a problem. So, and then we try to do the standardized method and control reagent are essential to improve reproducibility and confidence in the data accuracy. Yes. What is such kind of problem is that came from bias. Bias such as the understimulation of individual microbiota members, as well as contamination, depression issue that result in failed positive, negative taxon identification can be introduced at every step of microbiota analysis. Of course, the sampling, transportation, and the DNA extraction is very, very big bias is coming. So, and also library preparation, sequencing, data analysis. The bias are occur in every steps. And then finally, the result was changed or poor variability and the compatibility. And in our studies, we focus to DNA extraction and also library preparation and the DNA analysis. We try to make standard microbiome sequencing standardization, focusing to 3 parts. For example, this is a result that, for example, the operator A, B, C try to do different method A, B, C, so that I'm sorry, difficult, right? A, B, C percent, the method of A, B, C. So that if we use method A, operator B cannot get good result, but C is okay. Such kind of procedure is not so good with it. The method B is okay. Almost same people can get the same result used by the method B. This is okay. So, and also after the result, quite different using the method C or something. That is a problem. You can see the imagine right there. So, and also in the many papers already mentioned about that, the appropriate controls is important. The most important thing is that one of the reagent negative control and mock community should be included in the entire extraction, sequencing, and analysis processes, and their exact nature adapted to the specific study objectives. So quite recent papers, still there is mention about that. So, and then I heard that when we published the paper related to microbiome research, the referee requests to do, you should use, did you use mock community for evaluate your data or something like that? So, and then we try to make the standardization, the development of microbial characters and support the microbiome research. Yes, imagine like that. Yes, before develop of our research, I also communicate with researchers and also companies persons. This is a very detailed thing. Not remember that. The next slide, I show you that, but maybe what kind of mock community is necessary. Also, more useful one is better. And also the cover, the very wide microorganism is better. And also I heard the users, researchers said, require the quality. What kind of quality is important or something? So, and we got the information from users. So, and I try to do the best. We try to do the best. I show you. This is the first cocktail we developed, the general microbial cocktails using. Yes, this one is Bacillus bifidobacterium, Clostridium, blah, 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 blah. You can see that this, and also this one is a cocktail using cell cocktail and DNA cocktails. The 15 microbial species derived from human and natural environment. And all of this one is the microbes, all of the microbes should be biosafety level 1. And also diverse taxon, high GC, low GC, and also different genome size, because this is the very effect to the result of microbiome analysis. And gram positive, negative, and this is also affected by DNA isolation kit. And the DNA cocktail, the long size of DNA for long read sequencing. That is essential, the point to develop one. And until now, I'm already distribute from NBRC culture collections. The one is a very general one, and another one is we are much more focusing to human microbiome. So it means that there are different species. We use the different species, and this is a price. So maybe you can imagine that very cheap. So for example, yes, the less than 5,000 US dollar, yes. This one is very 200 US dollars. So, and then also in Japan, the user said how to use your cocktail. So, and then we show this slide. So the researcher can use for the reference our microbial cocktails mock community. So, and then this one is we collected the equal volume of 15 strains. This one, for human one, is equal cell number of human microbiomes, so that the result should be equal. But sometimes if the experiment is no good, so that the result quite different from real numbers. So if the same is comes, so that your experiment is okay, but then if not good, it's your data is not so good. You can do like that. You can use quality control. I explained to do that. And the second one is that sometimes we want to compare the data. From one institute, the second institute, we have a collaboration. That time we want to share the microbiome results. So that time, so please use our reference for the quality control of each researchers, right? I think yes, this one is that, yeah. In the first papers, we published the standard method for analysis of microbiome, this validation and the standardization of DNA extraction, the library construction method for metagenome-based human fecal microbial measurement. Sorry, we use our cocktails to make the standard method. It's do like that. And also you can imagine that, sorry, in Japanese, DNA extraction, we try to 21 conditions in 9 DNA isolation commercialized kit. And for library preparation, we try to do 28 conditions in 11 library preparation kits. We try to many method. So, and which is the which method is the best of the how to say the best of the yes, get the how to say the recommended standard operation method for microbiome analysis. At that time, we need the cocktails because cocktail means we put the equal volume, equal cell numbers here. Also, this one is equal amount of DNA was mixed in the here. The data should be equal. This is the data, one of the example. Yes, how to say, different analytical method, we try to do this one. So, and then accuracy is this method is best, but this one is no good. And also library preparation, that this is good, but this method is no good. So, and we should use this plus this or something like that. So, and then we try to make a standard. So many data are coming in the papers. So, and if you are interested, please read our papers. This one, and also we developed the recommended protocol. No, we never say SOP because we didn't apply to ISO method or something. Just we call recommended protocol. We deliver to recommended protocol the microbiome research group in Japan and companies, so that, and then also please try to do recommended our method and your method, so that if from A laboratory until J laboratory, they try to our method so that the result is almost same. But if the different method, they use different methods, for example, X group and Y group try to their own method, so that results are quite different like that. So, and then we think that our recommended protocol can be used in the variable researchers. Sorry, please. So, yes. And also, somebody want to know how different our cocktail with commercialized one. This is summarized. Maybe this is correct, I think, summarized. Until now, in the world, you can receive the microbial cocktails from ATCC and the Zymo Research Company. And NBRC is a little bit difficult because now we distribute to domestic. And another one is the NIBSC and also the NIST. The ATCC provide microbial cell cocktails and DNA cocktails. Zymo Research also both. NBRC also both. And NIBSC, this one is will be can be used for the standard of WHO supported by WHO. That one is provide only DNA cocktails, and the NIST is provide human fecal materials for reference. So in the next slide, I will explain about how to make our microbial cocktails. The time is okay. Maybe okay. So yes, this one is 10 days. Yes, 10 days before the method was public. that we use HPLC-based method for counting the genome copy number of cells, allow the production of a high-quality mock community of bacterial cells. So I cannot say strong, big voice, but also we buy the cocktails from commercialized one, and we compare with our cocktails and how different. So that, yes, I think I want to say that our cocktail the quality is higher than others, but based on our analytical. So because you can imagine that it's easy to mix the equal number of cell. Maybe you can imagine or not. Or also fast we think that, but very difficult to make the same amount of cells. So first we try to flow cytometer to calculate the number of cells. And the next one also, we try to microscopic analytical method. First, in the first trial, we count the cell number based on microscope or flow cytometers. But I think some kind of bacteria is combined. So and very difficult to collect numbers of cell. So I'm sorry, the color was changed. This is theoretically the equal volume we try to make, but when microscopy we count, it's maybe we cannot make same amount of cells. Also flow cytometer. So and then we think about another method. Yes, here. Yes, I should explain. We focused on the method. This is the original one. Is not published. Is not our method, but some researcher published the measurement of DNA using HPLC to quantity the amount of adenine in bacterial cells. The principle is the treating nucleic acid with unreleased adenine and guanine, which are prime bases. You know, I'm sorry, I mistake. Mistake. Yeah. Here, the cell is also acid treatment by these conditions, and then so that the adenine and the guanine is released. So we use the known microorganisms. Known meaning is that we got the whole genome sequencing. So and we know that if one cell, how amount of adenine are there in the cells, we can calculate from whole genome sequencing. So and then after treatment by hydrochloride, we apply to HPLC and calculate adenine, so that this amount of the data we compare with whole genome sequencing, the theoretical amount from the whole genome sequencing. So and then we can make we can set up the equal number of cells to make mixture. So and then we try to do that. Sorry, this is the color becomes same. The HPLC method is almost same with theoretical amount. And also this line is previously I said to you, the request from the researcher or microbiome researchers, they need the accuracy. They need the level of accuracy, the less than this and less than this. But this is our HPLC method is the best. Better than other calculations. This is also, yes, everything. I'm sorry that this is skip. So and also we are also another research. We try to ask the evaluate of our cocktails to our in Japanese researchers. Also, this one in this paper, we discussed the high quality of NBRC created mock community and the effective of recommended protocol for microbiome measurement that we provide. Yes, almost same. Yes, this is a very small target, but also so many people are involved with this research. So, and then based on this, our result in Japan, almost, not almost, but many microbiome researchers are following our protocol. Thank you very much for attention.